those of you that have late work, obviously. Many of you have already done the work and have turned it in on time. So DNA transcription. What do you need to know for the exam? What you need to know for the exam is that you got this thing called DNA. It has a double helix, right? It's badly drawn there, so let me redraw it. So you have a double helix. I showed it to you in a video, in a kind of an animation. Jeez. I showed it to you in an animation, correct? All right. There is a section of the DNA that we say begins. This is where transcription begins. What is transcription? Making DNA into RNA. And there's a section that where transcription stops. This section, from where transcription begins until transcription stops, is called the what? This section of DNA? A gene. Correct. That is a gene. There is a portion of the DNA that acts as a regulator. This portion of the DNA here is where transcription factors bind. Transcription factors are the thing that do what? What do transcription factors do? Go ahead and say whatever it is you're saying. I can't hear you. They're the result of signals. You are correct. It's the signals, it's the cell signals like the hormones like estrogen, testosterone, insulin. Those are the signals that send via a pathway of changes of chemical reactions that create these little factors that get on the DNA. The factors act as a landing strip. So they create a place for something to land. What's the thing that's going to land? What's the enzyme that does transcription, that takes DNA and makes RNA, a polymer of RNA? If your back is to me and I'm drawing on the board, I really don't. In fact, I'm having a conference with both of you right after class. And I'll send them. I've asked you several times not to have your back to me. Yeah, we'll have a conference with you. We'll call your mom after this and we'll figure this out. But right now I'm telling you, turn around and face forward. So what is then the enzyme that's going to take DNA and make RNA? Huh? That's right. It's on the board. It's a thing called RNA polymerase. RNA polymer ACE because it's an enzyme, and polymer because it's making a polymer. Polymer what? RNA. RNA polymerase. How does the RNA polymerase know that that's the beginning of the gene? It knows that because of the transcription factors. How did the transcription factors end up binding to this portion of the gene? Why this gene and not some other gene? Because it had a signal. What was that signal? Who knows? Calcium, some kind of hormone, something told that, and there's several different signals that can be used, but some cell signaling mechanism told this cell that this gene needed to be turned on. The example we used in class that you will see on the test, again, is insulin. Repeatedly, I've used insulin throughout this whole semester. So there should be no question that that's coming. So that insulin gene signals the start of a transcription or the making of mRNA. So the result is that you get a copy of one of the two strands, remembering that this RNA polymerase has to unwind the DNA, has to read it. You remember the video I showed you of it. You can go look it up on Google again if you'd like. You just have this repeated chain of RNAs, right? Where you had if the DNA was GTAT, GCAT, then your mRNA, and I'll go ahead and put it in yellow, is going to be CAUACGUA. One of these two sides, one of the two strands is going to be red, and it's going to make this thing called, we call it well, it's not mRNA yet. It's called RNA. RNA. Now we got to get this RNA. Why do we have to do this? Why do we have to have an enzyme, or why do we have to take this DNA and turn it into RNA? Why is this even necessary? And that goes back into where DNA is. Where is DNA in the cell? It's in the nucleus. It's surrounded by a membrane. The DNA is here in the nucleus, in eukaryotes. It's surrounded by a membrane. So you have to take that DNA, that message, and get it out of the membrane. The DNA will not pass through that membrane. It's too big. It won't leave. But if we took a close look at the membrane, then what we would see is the membrane has these pores, these holes. We call them nuclear pores. Looks like Swiss cheese. And these pores allow what to come out? RNA comes out of them. And things go in. And they go from the nucleus, they go into the endoplasmic reticulum, which is a big membrane that's connected to the membrane. It's connected to the nucleus. And on the outside of the rough endoplasmic reticulum is what? Ribosomes. And it's these ribosomes that are going to make the protein. They're going to do the translation. They're embedded in this membrane. This is a membrane. It's hard for me to draw. I've shown you. You have really great drawings in your study guides. I've shown them to you in class. We can show them to you again if you need to see them. But this portion of the endoplasmic reticulum is, endoplasmic reticulum, ER, is rough. It's rough because there are ribosomes on it that are making proteins. Well, where is it getting the message? What order of amino acids to put where? How do you get the primary structure from the ribosomes? You follow the message. Where's the message coming from? The DNA originally, correct? But it's mRNA that's the messenger. That's why it's called mRNA, messenger RNA. Now, interestingly, this is an interesting part because it kind of refutes the whole one gene, one enzyme, the what we call the central dogma of molecular biology. What it is you have this DNA, this gene. I'm going to go ahead and draw it out this way. It's just a line, if that's okay with you. If that's the gene, right? That's the beginning of the message here. And this is the end of the message here, B and E, right? When you have the mRNA, once you get this RNA is then cut up. It's cut up. And different sections called introns are cut out. They're gone. And then those other sections, the leftover sections, are put together. So what I'm saying to you is, if the message were C, if the message were, oh my God, are you kidding? Were C, A, U, A, C, G, G, U, A, U, A, C, U, C, all right? If that was the message that came from the DNA, your cell might, it depends on evolution, what decisions made depends on the conditions. So what the message you'll get once it's mature, once you've cut out, once you've done your what we call post-transcriptional. Let me just write it. Let me erase it and just write it on this one. What we like to call post-transcriptional modification. It sounds fancy, doesn't it? It's just, all that means is, post means after what? Transcription. And what are we going to do after transcription? Modify it. What modification are we doing? What changes are we doing? We're taking out the what? The introns. We're just going to take them out. And we're going to put them together. And we got C, A, U, and we don't have ACG anymore. We have G, U, A, and then we have C, U, C. So this is now the mature RNA. There's some other changes that happen as well, but we'll stop there. So now you have the mRNA. This is what's going to be made into a protein. Are we clear? So the message started off as DNA. It started off as G, T, A, T, G, C, A, T. Then it got turned into C, A, U, A, C, G, U, A, and whatever come after that. Then another enzyme cut out the introns and spliced together the rest of the mRNA. And now you got what's called your messenger RNA. What's the job of this mRNA? To send a message. The message from the gene to what? To what part of the cell? What part of the cell is going to use? Somebody said the nucleus. No, the nucleus already has the DNA. It's in the DNA. The message is in DNA in the nucleus. Let's go over it one more time, and then we got to move on. The message is in the DNA in the nucleus. You first change it to RNA. Then you cut out the introns. Now you have your mRNA plus some other changes. You do that in part so it can come out of the nucleus through the nuclear pores and go where? To the endoplasmic reticulum, the rough one, because what's in the endoplasmic reticulum? The ribosomes. Ribosomes are made of RNA themselves. Another gene made ribosomes already, made the DNA from ribosomes. So then what? Ribosomes are going to do what? What's the whole point here? Make proteins. So you're going to have two parts. You have a big subunit and a little subunit. This is a ribosome. It's made of two subunits, two parts, the big and the small. I'm not going to tell you. We can go into whether it's an 80S or 30S or whatever. Let's focus on two subunits. Are we clear? Mm-hmm. And there's 3 spots in it. There's 3 spots in the ribosome. 1, 2, and 3. 3 spots. 3 attachment points. The mRNA comes in on one side. And it's going to read every 3. It's going to read these messages 3 letters at a time. In the case that we have here, it's C-A-U. Then it's going to read what? G-U-A. Where am I getting this? C-A-U, G-U-A, C-U-C. Then it's going to read C-U-C. and whatever else is in that mRNA. Are we clear? The very beginning of every protein, the very beginning, every mRNA has, is this. I'm sorry, that's not there. Let me erase this and make it a little closer. No more room here. I need more room. The beginning of every messenger RNA are three, what we call codons. These three are called codons. C-O-D-O-N, codons. The very first codon is A-U-G. There might be other stuff on here before it, but this ribosome won't do anything until it sees what? What is it? What does this ribosome need to see in order to start? Nope. It needs to see RNA. Well, you're correct on that. It needs to see a messenger RNA. But what part of that messenger RNA does it have to read in order to start making a protein? What three codon? What codon? What three letters? A-U-G. 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 Every mRNA has it. This is what we call the start codon. There's only one start codon. Are we clear? A-U-G is the start. That's it. AUG starts. So as soon as that ribosome sees AUG, now it's going to start doing what it does, make a protein. Now I left some spots open there, didn't I? Did you see those three spots? All right, so what do you think is going to happen at those three spots? Go back. Let me take this. Let me copy it, move it over. So let me paste it. So once we have, if we get rid of all this, right? We have a ribosome, so we know it's a ribosome. We know there's codons. We know the codons are on the mRNA. We know there's two parts of the ribosome, big and the small. It's a complex. There's a bunch of proteins, mRNAs or RNAs. These are the codons here on the mRNA, codons. There is an opposite matching pair. Don't we do that? Isn't that a theme we've been doing all along? DNA, ATG gets matched to TAC, right? We do that. We did it when we transcribed from GTA to CAU, right? We did that when we made the mRNA to begin with. We keep matching. They keep base pairing. We call it base pairing. We have three. Each codon is going to have a base pair. What do you think we call that? The opposite of the codon. You learned the opposite when we said we didn't call it parallel. We call it what? Because it's the opposite. We call it what? Anti-parallel. So this is not the codon. This is the opposite. So we call it what? Anticodon. Anticodon. The opposite of what you have. So if you have AUG, what are the three that you'd expect to see in this ribosome? Say again? I'm sorry, I can't hear you. UAC, right? UAC is correct. That's what you'd expect to see. Well, where is that UAC going to be? Because it's matching, and what is it? What do you think you need to? What are you trying to build again? Remind me. A protein. What is a protein? A bunch of amino acids put together in a specific order. What's the order? Well, there it is. This is the order. These are the codons that tell the ribosome what order. So this is the anticodon that's matching. But so far, all we're talking about are nucleotides. Where's the amino acid come in? Where is the amino acid? I'm going to show you. There's a bunch of these in your cytoplasm. They're in the cytoplasm. We call them tRNAs. They're another type of tRNA. They're another type of RNA. That's right. It's a transport RNA. To me, it also looks like a T, but almost like a ghost T or whatever. Attached to this tRNA on this end is an amino acid. In this case, let's go ahead and use methionine. We have 3-letter codes for them. That's your methionine. On the, that says what? MET. MET for methionine. Are we good? It's a lot, but I think it's not too hard. It's not conceptually hard. If you remember, the DNA's in the nucleus has to get out. The message has to get out. We have to change it to RNA. If you remember where you had a T, you're going to have a U. You're going to replace T with U. If you remember that A goes with T and G goes with C, or A goes with U and T goes with A, right? If you remember all that, and you remember that you got to cut out the introns, they get left out, and you splice them together. Remember that. Now you got an mRNA that goes into the ribosome that's in the cytoplasm. Where's translation happening? I'm asking you the questions I'm going to ask you next week. Where's translation? This is translation. Where's this happening? RER, where? Where in the cell? Is it in the nucleus? No, it's not. What's in the nucleus? DNA. Where are the ribosomes? In the cytoplasm that's in on the RER or free floating in the cytoplasm, but they're in the cytoplasm. We all agree on that? Ribosomes are made of RNA and proteins. They have with them 3 spots. The the start codon is known as AUG. These are the bullet points that you need to know for next week. So I'm telling you what you need to know. I'm telling you what I'm going to ask you. So what do you think, if this is carrying methionine, what do you think is on the other end of this tRNA? These tRNAs are in the cytoplasm, in the endoplasmic. It's the anticodon. Just because we're running out of time, I'm going to answer for you. You have UAC on this end. So the ribosome, you remember that RNA polymerase, the video I showed you, a ribosome? I mean, of an enzyme that reads the DNA and makes that long mRNA really quickly. Ribosomes do the same thing. Say what? Huh?
Like this drawing?
Yeah, like that one, that drawing, that's correct. Well, this is going to happen, and this is the way it's going to happen. It's going to come in. The ribosome is going to match up, and look what happens. They match up. They base pair. Did you see that? They base pair. What does that mean? U matches with A with U, G with C. You see that? Then it clicks on over. There's 3 spots to this, so it clicks on over. And I'm going to go ahead and move it up a little closer, if you don't mind. Ooh, I don't know what just happened there. Let me connect all this. So what happens is that this ribosome takes, and you have these 3, UAC, that has this tRNA on it, right? This thing has methionine. Then the next one slips in. You have your mRNA here. And your next one's CAU. There's another tRNA. Each tRNA carries, each triplet code carries a specific amino acid. What is the next anticodon going to be? I can't hear you now. GUA, you say. GUA. Oh, I don't know why I put UUA. GUA. Why did you say that? Because C goes with G, A goes with U goes with A. Correct. Yeah? I would rather you not, but if you have to, go. So GUA happens if you have a tRNA that has the anticodon GUA, then you have an amino acid called valine, VAL. And inside the ribosome, you bond them together. In the ribosome, you make a bond, a peptide bond between them. That's what this is all about, right there, creating the primary structure. Then the next one that comes in as it keeps ratcheting out, right? I'm going to go ahead and draw it again. And of course, there's a second one, right? It says 2 subunits, so I don't want you to make a mistake. And so there you have your code. You have AUGs over here, and you got CAU, right? And then you have, what's the other one? GUA, right? No, that's not GUA. CAU, then, yeah, GUA. And then CUC. So now, here you got methionine, MET, connected to valine, right? Isn't that what we said? Yeah. Now we're connected to valine, VAL. Then you're going to connect it to a third one that's going to be what? What's the third one going to be? It's going to be CAU, correct? That's going to be connected to tRNA that has this, and it's badly drawn, but I'm going to go ahead, because for the sake of time, I'm going to go ahead and leave it alone. Otherwise, if I had time, I'd redraw it. So there it is. So what amino acid does, oh wait, did I draw this? Methionine, CAU. I messed up. The CAU is actually histine, so I made the classic mistake. And then GUA is valine. And then that gets bonded together. And before you know it, you have this long chain, and it could be very long because mRNA can be very long. This long chain of amino acid, connected 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 amino acid, et cetera, et cetera, et cetera, et cetera, right? Each different amino acid having different qualities, folding itself because of hydrogen bonding, so that you can get an enzyme that looks like this, another enzyme that looks like that, another enzyme that looks like this, right? So each shape, you can get collagen that forms skin, right? You can get keratin that forms nails. You can get digestive enzymes. You can get tubulin, microtubules. You can get intermediate tubule, right? Intermediate filaments. You can start building a cytoskeleton. Now you can start to make these proteins. Each protein doing a different function. How do you get different functions, different proteins? Because you have different messages in different parts of the DNA. They get turned on in different times, depending on the conditions that the cell is in when there's time to make them. Yeah, question?
I was asking, does it do the same thing for your hair?
Yeah, the same thing with hair. That's how you can inherit curly hair or straight hair or wavy hair. You get the different hair textures, the different hair colors, all because you started with that DNA all the way back here in the nucleus. You started with the DNA, and it turned into an mRNA that left the nucleus, that went to the ribosomes, that made it, that, by the way, got changed on the way out of the nucleus. Then it met up with the ribosome and made this long chain of peptide bonds that we call a protein. And if you have a lot of cysteines here, you got really curly hair. You don't have a lot of cysteines, you don't have a lot of curly hair. Or that's just a simple example, obviously. With enzymes, see, it's much more serious, right? If you have the right enzyme, then you're producing insulin and everything's good. You have the wrong one, you don't produce insulin correctly, and you have diabetes, type 1 diabetes, right? So disease comes from malfunctioning of either the gene or maybe the post-transcriptional modification, or something's wrong. It could be any one of these steps that could be wrong that could lead to a disease. Is everybody okay with what we just said? So there is a universal code. All right, so the question then becomes, how did I know that AUG was methionine? How did I know that GUA was valine? How did I know that? First of all, let's be clear. I did not memorize it. You have a periodic table in chemistry, you have a universal code. This is the code of all life on Earth. All life on Earth does this. Thank you for getting the door. I don't know who it is that's at the door. There's early college playing around? Unbelievable. So here we have what's called the universal code. This is, every creature uses this. This is what's amazing. And some more evidence about the thought of evolution, right? Think about it, the fact that bacteria use this, humans use this, trees use it, plants, algae. Every form of life on Earth uses this code, the same code. It kind of connects us all. So what do we find when we get to AUG? Who can find AUG? Let's A is what? AUG, let's take a look at it. Let's write it out. AUG. What's the first letter? A. And what's this? The first letter. So it's A. What's the second letter? U. And so A and U. So it's in this column, right? In this row, A and U is found in this box somewhere. And what's AUG? It's methionine, MET, also known as, let's be clear, start. Are we good? All right. Well, how do you know, how does the ribosome know that it's time to end? The end is not the last bit of the mRNA. The end is before the mRNA. When does the ribosome stop making a protein? Look on this chart and tell me when the ribosome stops making a protein.
AUA.
AUA. Wait, why AUA?
I said.
AUA is here. That's isoleucine.
I said U.
Wait, what? UAA is stop. UAG is stop, and UGA is stop. Those are your three stop codons. This is where you stop, guys. This is where the ribosome knows how to stop. So let's be clear. I should be able to, on the test, give you... Oh, by the way, this is a chart. You can make one for anticodons, but this is a chart based on the codons. Is that clear? What I mean to say is, if I give you DNA, this is what I'm going to do on the test. I'm telling you what I'm going to do on the test right now. You're going to have A, T, G, C, A, T, whatever, right? It'll be a different sequence. Then I'll ask you, what protein does this make? What protein is made with this code? What series of amino acids does this code make? And I'm going to ask you to ignore introns. I'm going to ask you to ignore introns, right? And I'm going to ask you just, look, here's this DNA. What amino acid sequence? I'm going to give you this chart. Obviously, you'll have this chart. Let's see if you can do it right now. Can you do it? Try it. Tell me, what's the series? Now, tell me. Do it. Do it on paper. Do it right now. Don't do it out loud. Do it right now. If you can do it. And then I'm going to do it together. I'm going to do it. And you see if we match. And if we don't match, let's see where you made your mistake. Do it. Make an amino acid sequence. What's the first thing you got to do? This is DNA, right? How do I know it's DNA? It's labeled DNA.
That's what I was thinking.
And then what else? STs, right? So it's clearly DNA. That's in the nucleus. You need to get it out. We're going to ignore the intron step, the post-transcriptional modifications. We're going to go right to making the protein. So you go, first thing you have to do is what? What is the first thing you have to do if you have DNA? turn to RNA. You have to do transcription. Correct. Excellent. I'm glad somebody's listening. So you have to do T goes to what? A. Then A goes to what? U. Then
U, A, C, G, U, A, C, G, U, A, G,
oh. Go ahead.
A, U, A.
There it is. Right? All right, so what's the series then of amino acids we have then? We have to find the start. And it's a 3 code, so we have to find the first AUG, which is what? The first one right there. Then what do? I have to read every 3, correct? So 3, the triplet code. 3, 3. So how many amino acids do I have here in this list? We're gonna assume it keeps going, but we have what we have, right? So how many amino acids do I have? 5. Okay. Well, what's the first one? What's the first amino acid according to this list?
Methane start.
MET, methionine, correct. Is then connected to what? What's it peptide bonded to? What's UAC?
I can't either.
I can't either. Let me, hold on. I'm sorry.
It's TYR.
TYR, that's right. Tryptophan, right? TYR. It's okay. You don't have to know the names because you're going to read the chart, all right? Nobody's asking you to memorize names. Let's be clear on that. TYR. Then it's connected to what? The next 3 is UAC, right? Am I right? UAC was, I messed up the, I'm sorry, I messed this up. Next one's GUA. GUA is what?
Val. Like Valerie.
Oh, I hate this. My menu's so GUA is valine. Then what's the next one? CGU, which is what? ARG. Do you see how easy this is?
Yeah.
It really is easy. It's going to be on your end of course exam. It's really easy to read. Yeah, there are some deep implications, but the stuff is easy. It's just matching it. Yeah.
So you're gonna give us one of those charts, you can have to memorize them.
Oh my Lord! I
He
said
said he was giving us a chart.
Oh, I didn't.
I love you, but you don't. You guys,
My friend
that
today.
I would never. First of all, I'm hurt that you would think that I would have you memorize a chart like this. Am I that awful that you all think that I would have you memorize this chart?
Yeah.
That just seems like, wow, you must think I'm a monster. All right, so anyways, there's 1, there's 2, there's 3, there's 4, there's 1, 2, 3, and 4. What's the last one here? AGA. AGA. AGA.
It's R.
It's arg. It's arginine. That's correct. Look at that. You can repeat. Did you notice that? No, there's a couple other things you need to notice, but we can talk about that after break. The key is that you're able to do this. You're able to know what is the amino acid. Is it time to go? All right. Are we all satisfied that you can handle transcriptions? At anyone in this room that has a problem, please speak up now or forever hold your peace, or at least for the weekend. Remember, all the work has to be turned in, any late work has to be turned in by the end of today. Remember, your final review, your semester final review is due Monday. You should have index cards made already. You had 2 days in class. God bless you and see you on Tuesday. If you have questions, see me Monday after your first final. If I'm here all day till 3:10. So if you have problems, see me on Tuesday during the day. Remember, after 12, you get to go home, but you can come in here and get tutoring if you absolutely need it, if you're confused about something. Have a great day.